Hi guys, welcome back to the Road to Success MCAT test prep series. In this video, I will be discussing the reproductive system and how it is important to biology. Now, this is a very important subject as it basically covers everything you need to know moving forward. I will be starting off by talking about mitosis and meiosis and their similarities and differences. I will then move forward with an anatomy lesson about the male reproductive system and the female reproductive system, and how sperm and ova are formed. I will end this video off by talking about the menstrual cycle, which is a very important and can be a very complicated task to learn. That being said, I'll break it down and I'll make it very easy, or at least digestible for you all to learn. We have a lot to cover today, lots of anatomy, lots of various different cycles. So let's get started and jump into mitosis. All right, let's start off with the cell cycle and mitosis. Starting with the cell cycle, we'll start with the concept of interphase. Interphase is the G1, S, and G2 phases of the cell cycle, and it's the longest part of the cell cycle, which is about 90% of the time the cell will be existing. There's also a G0 stage where the cell is simply living and carrying out basic functions. No preparation of division is occurring during this stage. During interphase, individual chromosomes are not visible within light microscopy because they're in a less condensed form known as chromatin. This is because the DNA must be available to RNA polymerase so that genes can be transcribed. During mitosis, however, the DNA will be condensed. So starting off with the G1 stage, the cell will create organelles for energy and protein production, while also increasing its size. In addition, passage into the S phase is governed by a restriction point. Certain criteria, such as containing the proper complement of DNA, must be met for the cell to pass the restriction point and enter the synthesis stage. During the S stage, which is for synthesis, the cell will replicate its genetic material so that each daughter cell will have identical copies. After replication, each chromosome consists of two identical chromatids that are bound together at a specialized region known as the centromere. Now I'll take a quick break and talk to you guys about the difference between chromatids and chromosomes. So the initial, one part right here is known as a chromatid. And two of them together like this is a chromosome. Lastly, what connects them, those little circles right there, we're going to call centromeres. All right, now that we got that out of the way, I only wanted to stop here because we're going to be talking a lot about chromatids and chromosomes moving forward. But with that out of the way, let's talk about the ploidy of the cell in the S phase. The ploidy will not be changing, even though the number of chromatids has doubled. But so in other words, in humans, we'll still have 46 chromosomes, but we'll have 92 chromatids. The G2 phase, the cell will be passing through another quality control checkpoint, where the cell ensures that there's enough organelles and cytoplasm for the two daughter cells that will be made. Also, the cell will check to make sure that DNA replication occurred properly, so that there's no errors or mutations that will be passed on to the progeny. Now lastly, let's talk about mitosis. There are 4 stages to mitosis, referred to by students as PMAT. So make sure you memorize that that acronym right there, starting with prophase. This is the most complex one, so bear with me. Prophase is the first phase in mitosis, and it involves the condensation of chromatin into chromosomes. The centriole pairs will separate and move towards opposite ends of the pole. Once the centrioles move to opposite ends, they'll form spindle fibers, which are made of microtubules. Some microtubules will form asters that anchor the centriole to the cell membrane, while others will extend toward the middle of the cell. Lastly, the nuclear membrane dissolves during prophase. The nucleoli might be less distinct or may disappear entirely. In metaphase, the centriole pairs are now at opposite ends of the cell. Kinetochore fibers will interact with the fibers of the spindle apparatus to align the chromosomes at the metaphase plate. Note that they don't split here yet because that's what occurs during anaphase. The sister chromatids will separate and they will be pulled towards the opposite ends of the cell. Telophase and cytokinesis occur immediately after each other. Telophase is the reverse of prophase, where the spindle apparatus will disappear while the nuclear membrane will reform and the nucleoli reappear. Chromosomes uncoil, they'll resume their interphase form, etc., etc. Each of the two new daughter cells will have a complete copy of the genome identical to the original genome that was present. Cytokinesis is a separation of the cytoplasm and organelles. This gives each daughter cell enough of material to survive on their own. Each cell undergoes a finite amount of divisions before programmed death. So for human cells, this will usually be about 20 to 50 cycles. After that, they can't divide. One thing I do want to talk about is that mitosis occurs in somatic cells only. And that's important because our next topic is meiosis. Meiosis is a bit different from mitosis in that it's 2 rounds of division as opposed to 1 round. And it also creates haploid cells or N cells as opposed to the 2N diploid cells. These occur, as you can see right here, it occurs in sex cells. And homologous pairs, or homologous chromosomes, pair on the metaphase plate as opposed to sister chromatids. Now, let's talk about meiosis 1. Meiosis 2 is very similar to mitosis, so I'll just write here that's aka mitosis. Meiosis 1, it's very, very different. So in prophase 1, the chromatin will condense into chromosomes and the spindle apparatus will form, and the nuclei will behave similar to how it did in prophase of mitosis. But the major difference here is that homologous chromosomes will come together and intertwine in a process that's known as synapsis. Each synaptic pair will contain 4 chromatids, and they are referred to as a tetrad. Homologous chromosomes are held together by a protein or a group of proteins called the synaptonomenal complex. Sorry about that, it's quite the tongue twister. Chromatids of homologous chromosomes break at the point of contact, called the chiasma, and they exchange equivalent pieces of DNA. This process is known as crossing over and is characterized by crossover events that occur on one strand of DNA. Note that crossing over occurs between homologous chromosomes and not sister chromatids. If it happened on sister chromatids, it would cause no change because they are identical. Now, I want to point out that crossing over occurs on prophase 1, and it's part of Mendel's second law of independent assortment. So I'll write here, second law, crossing over. In metaphase 1, the homologous pairs, or tetrads, will align on the metaphase plate, and each pair is attached to a separate spindle fiber. In mitosis, each chromosome is lined up on the metaphase plate by 2 spindle fibers, while in meiosis, homologous chromosomes are lined up from across each other and are held by 1 spindle fiber. During anaphase 1, homologous pairs separate and are pulled to opposite poles by the cell, or opposite poles of the cell. This process is called disjunction, and it accounts for Mendel's first law. Sorry about that, it sort of overlapped. Here, I'll rewrite it again because I don't want to cover up the diagram. And this is the first law. During disjunction, each chromosome of paternal origin separates from its homologue of maternal origin, and each chromosome can end up in either daughter cell. This separation of the two homologous chromosomes is also referred to as segregation. Lastly, telophase 1, a nuclear membrane, forms around each new nucleus. It's essentially very similar to how telophase occurs in mitosis. After that occurs, the cell will divide into two daughter cells by cytokinesis, and in between meiosis 1 and meiosis 2, there will be a short rest period called interkinesis. That being said, meiosis 2, I already discussed, basically saying that it's like mitosis, so we don't need to cover that again. Moving on, let's talk about the male reproductive system. One thing I want to start off with is by saying that the male chromosome, the male sex chromosome, is a Y, while the female sex chromosome is an X. So, that being said, let's move straight into it. You can see right here there's a lot of anatomy going on, and you can study this on your own time, but I will want to focus on a pathway called 7-up, which is essentially the sperm pathway from your testes all the way exiting out the urethra. So, I'll say this slowly, but it starts off with this in the seminiferous tubules. I don't believe it's labeled on this one. But that being said, you can study the anatomy on your own time. Please focus on just the pathway itself. The seminiferous tubules then go into the epididymis. Then after that, it'll go through the vas deferens, traveling all the way across here, up to the ejaculatory duct here. After the ejaculatory duct, N stands for nothing. And then it'll pass through the urethra, right here, and then it'll exit out through the penis. I know I went a little bit fast there, so I'll say it one more time slowly. Seminiferous tubules, into epididymis, to the vas deferens, to the ejaculatory duct, to the urethra, to the penis. That being said, let's jump into the process of spermatogenesis. The formation of haploid cells through meiosis. In males, diploid stem cells are known as spermatogonia, and after replicating their genetic material in the S stage, they will be developing into diploid primary spermatocytes. The first meiotic division will result in haploid secondary spermatocytes, which then undergo meiosis 2 to generate haploid spermatids. Finally, the spermatids undergo maturation to become mature spermatozoa. Spermatogenesis results in 4 functional sperm for each spermatogonium. I want to point out that mature sperm is very compact. They consist of a head, which contains all the genetic material, and a midpiece, which serves as a connecting point and ATP generator for the tail. I also want to point out that each sperm head is also covered by a little cap known as the acrosome. The acrosome is derived from the Golgi apparatus, and it's necessary to penetrate the ovum. Once a male reaches sexual maturity during puberty, about 3 million sperm cells are produced every day. And we'll be doing that. Men will be doing that for over the course of their lifetime. So from 13 on or 12 on, whenever you hit puberty, men will be creating sperm for the rest of their life, and it'll be healthy as it was from day one, or not day one, from when you were 13. Moving on to the female reproductive system, there's a lot less anatomy here, but there's a lot more complicated stages, so bear with me. So I'll just talk really quick about the pathway of an egg. So they're created in the ovary, and they're eventually ejected and go through the fallopian tube, where they'll eventually be implanted in the uterus for fertilization and growth. I thought I had a little bit more for you guys, but that's it apparently. So moving on, let's talk about the process of oogenesis. By birth, all of the oogonia have already undergone DNA replication, and they're considered to be primary oocytes. These are 2N, like their primary spermatocytes, and they're arrested in prophase 1. So all of these primary oocytes are in prophase 1. After menarche, or the first menstrual cycle, one primary oocyte will be released every month to complete meiosis 1, producing a secondary oocyte and a polar body. The division is characterized by unequal cytokinesis, which unfortunately the polar body receives none of the main organelles. The polar body eventually will be just atrophied. It will not serve any major purpose. The secondary oocyte, on the other hand, will stay in metaphase 2, and it will not complete the remainder of meiosis 2 unless fertilization occurs. So I'll just write it here, metaphase 2. The last phase I want to talk about is the menstrual cycle. This is going to be tested heavily on the MCAT, so please pay attention to this section. The menstrual cycle can be broken down into 4 different stages. Starting with the follicular phase, this is where menstrual flow sheds the uterine lining of the previous cycle. GnRH secretion from the hypothalamus increases in response to the decreased concentrations of estrogen and progesterone, which fall off towards the end of the cycle. The higher concentrations of GnRH cause increased secretions of both FSH and LH. These 2 hormones will then work together to develop several ovarian follicles. The follicles will then begin to produce estrogen, which has negative feedback effects and causes the GnRH and LH, as well as FSH, to fall off. Estrogen stimulates regrowth of the endometrial lining, stimulating vascularization and glandularization of the decidua. So I know there was a lot that I just said right there. Bear with me. I'll write down basically in shorthand what happens here. So the increase in GnRH will cause an increase in FSH and LH, which will then cause an increase in estrogen, which then serves as a negative feedback loop, decreasing GnRH, which then leads to a decrease in FSH, a decrease in LH. Follicle stimulating hormone and luteal hormone. I'll be covering the specifics of those in a later video about the endocrine system. Anyways, the next stage is the ovulation stage. Estrogen here actually is pretty interesting in that it creates both a negative and a positive feedback loop. The developing follicles will secrete higher and higher concentrations of estrogen, eventually reaching a threshold where they will paradoxically create positive feedback, and GnRH, LH, and FSH will start to spike. The surge in LH is important as that is what is inducing ovulation. So I'll shorthand this as well. So the positive feedback loop here will cause an increase in estrogen, which will then cause an increase in GnRH, causing an increase in FSH, and a massive surge in luteal hormone, eventually leading to ovulation. Moving on to the luteal phase, the LH will cause a ruptured follicle to form the corpus luteum. This is what's going to be secreting progesterone. Progesterone is what maintains implantation, while estrogen regenerates the uterine lining. Progesterone levels begin to rise while estrogen levels remain high. The high levels of progesterone cause negative feedback on GnRH, FSH, and luteal hormone, preventing the ovulation of multiple eggs. I'm so sorry, I realized just here it says gluteal phase, not luteal phase. But anyways, it's luteal phase, and moving on towards the last step, the menstruation occurs. Menstruation, assuming that implantation does not occur, the corpus luteum will lose its stimulation from luteal hormone and progesterone levels will decline, and the uterine lining will slough off. Loss of high levels of estrogen and progesterone remove the block on GnRH so that the next cycle can begin. So I'll write just here, I'll finish it off because I already wrote it for these two. So increase in progesterone causes a negative feedback loop leading to a decrease in GnRH and a decrease in luteal hormone and FSH. Finally, right here, since there's a decrease in LH, there's going to be a decrease in progesterone. Which will eventually cause sloughing off, or menstruation. I know there's a lot of stuff here, but it's very important for the MCAT, so please study this slide. But that being said, that's all I have for you guys today. We covered the menstrual cycle, as well as the male and female reproductive systems. We talked about meiosis and mitosis, and we also talked about the cell cycle. This was a very dense passage, but it's very important for the MCAT, so I hope you guys were able to learn something. I hope I was helpful to you guys. That being said, I hope you guys have a great rest of your day, and happy studying.